This is Bio 182, Lecture 2.2 on microevolution. In the previous lecture, we talked about Hardy-Weinberg equilibrium and microevolution, and how Hardy-Weinberg equilibrium means that there is no change in the allele frequencies of a population over time. However, if microevolution is occurring, that means that we have a deviation from Hardy-Weinberg equilibrium and that microevolution is occurring. So in this lecture, we're going to talk more about microevolution. There are several mechanisms for microevolution. For the four main ones are mutations, genetic drift, or random death or survival, gene flow or migration, and natural selection. We're going to talk about each of these in more detail. So the first driver of microevolution is mutation. Mutations are where there are errors in DNA replication that can lead to mutations. Some mutations will affect the sequence of amino acids that the DNA codes for, with an array of different possible consequences for the individual. Some mutations can be lethal, so the organism carrying the new mutation will die. Some are neutral, meaning that the mutation is nothing that is beneficial or harmful, and therefore it doesn't affect the individual in one way or another. They can also be advantageous, where an organism that's carrying the new mutation has some positive effect on its survival or reproduction. So in the examples, we have mutations in Drosophila that result in different colored eyes. Or we have a single base pair mutation in humans that result in sickle cell disease. So sickle cell disease would be something that is harmful. Eye color in Drosophila could be beneficial if it somehow affects mating or sight and vision, or it could just be neutral if it's just the color of the eyes. In our class, we're going to talk about different types of mutations. So the first thing we need to talk about is point mutations which affect single genes. So in this case, we're talking about mutations that are affecting a single gene. We'll also talk about mutations that affect entire chromosomes. So point mutations affect one gene at a time. So in your normal DNA, you have your DNA that codes for mRNA. And if any changes occur in the DNA or the mRNA, it's going to affect the proteins that are coded for, and therefore can affect the building blocks of your cells and other things in your body. So a silent mutation is one where the mutated template DNA strand does not change the amino acid sequence of the polypeptide. So in this case, we have an A was changed to a G in the DNA, which causes it to code for a C in the mRNA. And so instead of, but that particular mutation is not going to cause any difference between the amino acids that are coded for. So in the normal DNA, it was coding for phenylalanine, and in the mutated DNA, it is still coding for phenylalanine because that particular sequence of DNA still codes for the same amino acid. So that one is called a silent mutation because although there's a change in the DNA, it's not affecting the amino acids. The next type of possible mutations in your chromosomes or in your genes is called a missense mutation. And this is where you have a mutation in the DNA that causes the mutation in the mRNA, and it does in fact cause a change in the amino acid sequence. So instead of that particular triplet coding for alanine, you're now coding for glycine. And so therefore you're going to have a difference in the amino acid sequence for that protein that's being coded for by that gene. The next possible type of mutation is called a nonsense mutation. And so in this case, you have a mutation where a premature stop codon is coded for. So anything that occurs after that mutation is no longer coded for in that gene, and that gene is now shrunk to the point of the stop codon. If it happens earlier in the gene, it's going to be a lot more harmful than if it happens at the end of the gene. But either way, anything that happens after that mutation that codes for a premature stop codon will then no longer be coded for in that gene. The next are called frameshift mutations, and so we call these indels because they are either insertions of DNA or deletions of DNA. And these are also going to affect anything that occurs after that point in the DNA because you are inserting or deleting a piece of DNA, and therefore everything in that frame is going to be shifted, and everything after that mutation will now be different than the original normal DNA. So we're at this point for nonsense and frameshift mutations, we are not just affecting one single amino acid, we're affecting everything that occurs after that particular mutation. Mutations can also affect entire chromosomes instead of just individual genes. So we can have mutations where an entire gene is deleted on a chromosome. So in this case, we have gene B in the sequence deleted. We can have a duplication event where gene B is duplicated. Those types of duplication mutations can actually be very important when we're talking about speciation events. We'll talk more about that coming up, but duplication events can be very important in when we're creating new species because now you have more than one version of that gene. You can have inversions and translocations, which can occur during crossing over during meiosis, where you have genes essentially being pulled out and either flipped around or moved from one chromosome to another. So an inversion happens on the same chromosome where your gene sort of just gets flipped around. And a translocation happens where a section of a chromosome gets moved from one chromosome to another. They sort of switch, flip-flop places. Chromosomal mutations can have very large effects when we're talking about evolution. In particular, gene duplication events allow an organism to have a spare set of genes so that one set can keep its original function and the other can mutate and acquire a new function. So this is a major force in macroevolution. And macroevolution means speciation events, which essentially is the creation of new species. When we talk about speciation, we're saying that we have a species that then splits evolutionarily, and you have a new branch where we've created an entire new species. And that's what we talk about during macroevolution. A well-studied example of gene duplication is in hemoglobin in vertebrates. So we've had two separate gene duplication events in hemoglobin that allowed the radiation from fishes to amphibians to amniotes. Because if you think about hemoglobin, we have lots of different types of cells in our blood, and being able to use that hemoglobin gene in a few different ways because we've had it duplicated, so it has its original use and now has two other versions that can have different uses. We are now able to have new species evolving because of that new array of different genes. The next the next driver of microevolution is called genetic drift. This is a change in the gene pool of a small population that is due to chance. So when we talk about genetic drift, we're talking about chance events that are affecting the allele frequency in the gene pool of populations. This occurs faster in smaller populations because if you think about a random event occurring, if you have a large population, even if 40% of a large population of individuals is affected by a chance event, you're still going to have a decent number of individuals that are not affected. However, in a small population, if you already only have a few individuals, if 40% of them is affected, you're going to have a larger effect than in a larger population. So an allele can be completely lost, reach a frequency of 0% or be fixed, so be 100% based on chance events. Genetic drift is a mechanism that has no direction, so it doesn't lead to adaptations. We just have chance events that are causing differences in the gene pool, and it's not based on survivorship or the ability to reproduce. It's only based on a chance event that has nothing to do with the ability of that species or those individuals to survive. And within genetic drift, there are two main patterns. The first is called the bottleneck effect, and the second is the founder effect. And we're going to talk about each of those in a little bit more detail. So in genetic drift, in the bottleneck effect, we have an original population, which is usually it's a larger population. We have a bottleneck event, which means that those individuals in that population, a very large number of them are not going to survive for some reason. It could be a flood or a tornado, something that wipes out randomly a large chunk of the population. And then the smaller population that survives is not going to have the same amount of genetic diversity that original large population had. So we start with a larger population with quite a bit of genetic diversity because of a chance event that wipes out part of that larger population and only a small number of individuals survive. Those small number of individuals are not going to maintain and bring with them the genetic diversity that the original population would have had. And therefore, you're creating a new allele frequency and a new genotype frequency in your surviving populations. This is particularly common in endangered animals like cheetah, which suffer from genetic drift where variation in their alleles is lost. So there are so few cheetahs left in the wild that their genetic diversity is very, very low because only a few individuals survived to then go on and create the next population. One of the most well-studied examples of a bottleneck effect in a species is in Florida panthers. And so for these individuals, Florida panthers are essentially the same species as pumas or mountain lions, but they were their own distinct population. During the early 20th century, the population dropped to a size of as little as 6 animals. And genetic analysis shows that this could have included just one female, meaning that the current population had just one ancestral mother and then 5 males. And so the genetic diversity of that population goes way down. And so because of that, there was a lot of inbreeding in the panthers that came from this ancestral mother, and it led to some individuals having kinked tails or depressed immune systems, and a very, very low genetic diversity. To try to remedy this situation, scientists translocated individuals from Texas to Florida to try to rejuvenate the gene pool. Translocation is a very common practice in endangered species to try to increase the genetic diversity of those populations, because without genetic diversity and a high amount of inbreeding, you are going to end up with individuals that have more and more genetic defects, and therefore your surviving individuals will have even lower chance of survival. A founder effect can also occur in humans. So when different groups of people were colonizing the Americas, the group that we call the Amish created a small island of inhabitants, and we say island in that they were sort of culturally isolated from others. And so in those individuals, the Ellis Van Creveld syndrome was the result of that founder effect, where it's a recessive allele that causes this, that became more prevalent in Amish populations because they were not breeding with anyone outside of the Amish community, and therefore it became more and more common through those inbreeding occurrences between members of the Amish community. The third mechanism that can affect microevolution is called gene flow or migration. Gene flow is the transfer of genetic variation from one population to another. If the rate of gene flow is high enough, then two populations are considered to have equivalent allele frequencies, and therefore they're effectively considered a single population. However, if there's complete disruption of gene flow between the two halves of a population, it can lead to speciation, which is a macroevolutionary process. So gene flow is affected by barriers between different populations of individuals of the same species. How big the barrier is determines the amount of gene flow between those distinct populations. So it could be an exchange of genes between two populations of the same species of birds that live on either side of a stream, where they have distinct allele frequencies in their gene pools. And then if one individual happens to make it over and successfully mate with individuals on the other side of the stream, you're going to start having a mixing of their genes. Gene flow becomes an issue when we're talking about endangered species, because in certain areas, if we think about farming, usually farmers are required to leave a certain amount of natural landscape in their farm fields. And that creates islands within their larger biogeographical area, where you have small groups of inhabitants that are isolated from others across large stretches of fields. This occurs in the Amazon. It also occurs in the Midwest, where farming is very, very common. And those barriers, depending on the species, can be large enough to create islands of populations that are not able to interbreed. When we think of, if you look at the picture on the bottom of the slide, when we think of humans moving around the planet, these are shipping lanes and road networks that move humans and other goods around the globe. And so when you have a lot of movement of humans, you have other things that come along with humans. You have plant and animal species that either intentionally or unintentionally sort of tag along with products and people that are shipped around the globe. And that is how we end up with invasive species, as well as a lot of mixing of the gene pools of humans as well as other animals on the planet. The fourth mechanism of microevolution is natural selection. Of all causes of microevolution, only natural selection results in adaptation. The other three cannot result in an adaptation. They can cause changes in the frequency of alleles in a gene pool, but natural selection can actually cause adaptation to occur, and the effects of all the other forces are random. But natural selection can be moving in a direction. So if before selection we have individuals that are anywhere from yellow to red on a color scale, selection can select for only those red individuals. Maybe they blend more into their environment. Maybe mates only seek out red color. And therefore, we're changing the allele frequency of that final population so that it is made up of only individuals with that certain allele that codes for a red color. And so in this example, we're looking at resistance to antibiotics, where certain bacterial cells do naturally have a resistance to antibiotics. And therefore, if we continue to use those antibiotics, those bacteria will flourish because they cannot be affected by that selective pressure, which is the antibiotic trying to kill those bacteria off. So when we talk about evolution, the thing that most people say immediately and first would be that Charles Darwin is an important part of evolution and natural selection. And you are correct in that. Charles Darwin did propose the first correct mechanism for evolution, which is natural selection. There were other people who had proposed mechanisms for evolution that turned out to be inaccurate, and Darwin was the first one to correctly describe natural selection. In addition, he proposed that all species of life evolved from one common ancestor. If we only look at evolution from this perspective, we're missing a lot of important information. So in the past 150 years, Darwin's idea of natural selection has been synthesized with several scientific fields like genetics, statistics, paleontology, geology, as well as others, and is now a fundamental part of the modern evolutionary synthesis, which continues to be refined, and it gives continued strong support for evolutionary theory. So the evolution is considered a theory. And if you think back to our first lectures, a theory means that it's not a hypothesis. It is something that has had many, in this case, thousands and thousands of studies that have supported this idea of evolution. And so we consider evolution to be a theory in biology, not a hypothesis. So one of the most important contributions that Darwin had to our study of our understanding of evolution was his description of natural selection and how it worked. And his use of and observations of finches in the Galapagos Islands were one of the things that allowed him to come to those conclusions about natural selection. So the Galapagos Islands are located off the coast of South America, and they have an ancestral seed-eating ground finch that lives on mainland South America. And over time, individuals from that mainland population were able to make it to the Galapagos Islands and found new communities there. The different islands within the Galapagos Islands have different environments. They have different trees and fruits and different things like that, different things for the birds to eat. And over time, all of these finches evolved from their common ancestor, but they have adapted to the specific environments on which they live on these individual islands. And so one of the main things is that they have beaks that are adapted to eating the types of foods that are on those islands. So they have adapted to the environmental constraints of their new surroundings. And that process of evolution occurs when you have an individual that has a heritable difference from other individuals. That individual has that adaptation that makes them better able to survive and therefore reproduce in that environment. And therefore, that individual's genes get passed on. And so because of that, because there was already genetic diversity in the beaks of these finches, when they arrived at the Galapagos Islands, they were able to radiate and speciate and fill in these niches all over these islands and specialize on the types of food that are there. So birds that have mainly insects as food on their island, they have beaks that are good for eating insects. Birds that have different types of seeds, different sizes of seeds, determine the size and the strength of the beak on that particular finch. Whether the bird has to dig into a cactus, they have longer beaks, and then birds who eat fruit have beaks that are well adapted to being able to eat that type of food. So these finches were able to go in and colonize those islands and utilize the resources that were already there. And because they had genetic diversity, they were able to utilize all of the different islands and all of the different food items on those islands. Another good example, which uses humans of natural selection, is human skin color. So if you think about skin color in humans, we have a very wide range of skin color from very, very fair to very, very dark. And the color of your skin is generally determined, and we're talking about the color of your skin in its natural form, not the color of your skin because of a suntan. So if we, in general, the skin under your underarm doesn't get a lot of sunlight, so that skin color is considered your natural skin color, the skin color that is underneath your arm. And what we see in humans is that there tends to be darker skin color near the equator, and as you move further and further away from the equator, you have lighter and lighter skin color. And so in the top set of graphs, graph B is showing that the reflectance of skin decreases towards the equator. So your skin is going to absorb more sunlight instead of reflecting it. The second set of graphs is showing that there are two different pigments in your skin. The first one is called pheomelanocyte, and it is a red or yellow pigment. And then the next one is called eumelanocyte, and that produces brown or black pigment. And your ancestry determines the amount of this pigment that you have in your skin. And so the more of that brown or black pigment that you have in your skin sort of creates a solar umbrella over your skin that prevents UV radiation from getting in. And that UV radiation, we don't want that to get into our skin because it breaks down folate, which is a B vitamin that's important for our body functions. So the closer you live to the equator, the more you are encountering high levels of UV radiation. So you have that darker skin tone to help protect that B vitamin that's in your skin. Another part of it is that humans need to make vitamin D in order to survive. And so if you have darker skin and you're not letting a lot of that UV radiation in, it's because we need UV radiation to make vitamin D. But if you live in an area near the equator, you're getting enough of that UV radiation all the time that you can go ahead and block some of it out. As you move further and further north towards the Arctic Circle, you're getting less and less UV radiation, and therefore you need to let in more of that UV radiation to your skin so that it will make that vitamin D. and therefore you have lighter skin up near the poles where you're getting less UV radiation because you don't need to protect your vitamin B as much from UV radiation, and you need that UV radiation to get into your skin to help your body produce vitamin D. Whereas near the equator, you're constantly being bombarded with lots and lots of UV radiation, and so your skin has that melanin to protect your vitamin B or your folate, and you're getting enough UV radiation on your skin at all times that you are able to block some of it out and still get enough to make vitamin D. So we've talked quite a bit about microevolution, and we will be talking more about macroevolution. A lot of people have an easy time learning and understanding microevolution, which is where we have changes in allele frequencies within a population over time. This is easily measured. It has simple predictions under different microevolutionary forces. The four mechanisms which we've talked about are natural selection, mutation, gene flow, and genetic drift. However, microevolution can accumulate, those changes, those microevolutionary changes can accumulate, and they can lead to macroevolutionary processes, which is evolution above the level of a species. And so this is going to be processes that include speciation and taxon extinction. So we do have quite a bit of evidence that macroevolution is occurring. The first bit of evidence is that there are similarities among living organisms. Those similarities are at the cellular and molecular levels. We can also see them in developmental biology as well as in anatomical structures. And then the second line of evidence is the distribution of living and fossil organisms in time and space. So at the cellular level, we can see similarities among living organisms, and we can see this in a lot of the postulates of modern cell theory. So all known living things are made up of one or more cells. All living things arise from pre-existing cells by division. The cell is the fundamental unit of structure and function in all living organisms. Cells contain DNA, which is found specifically in the chromosome, and RNA, which is found in the cell nucleus and the cytoplasm. And all cells are basically the same in chemical composition in organisms of similar species. So no matter what living thing we're talking about, these different parts of the cell theory are true for all living organisms. The second part of similarities among living organisms is at the molecular level. So we're talking about DNA and RNA. And so those, as well as proteins, are information molecules in that they retain a record of an organism's evolutionary history. The approach in comparing DNA and RNA is to compare the nucleic acid or protein sequences from different organisms using computer programs, which you guys are going to actually get to do in a lab, and estimate the evolutionary relationships based on the degree of similarity between those sequences. So in other words, closely related organisms will exhibit fewer sequence differences than distantly related organisms. So an example of this would be that we used to consider birds to be a separate lineage from mammals, reptiles, amphibians. But we now know that birds evolved from a lineage of dinosaurs that were the sister group of crocodiles, and therefore they can be put within the reptile clade in biological organization. Another part of molecular evidence is that all living organisms use DNA or RNA for their genetic information. And therefore, at that level, all organisms are using those same bases and base pairs to store their genetic information. Another type of similarities among living organisms is seen through developmental biology, where a lot of individuals look very different as adults, but they look very similar either as embryos or as juveniles or adolescents. So we know that whales evolved from an ancestor that was a hoofed mammal and it had teeth. Toothed whales have full sets of teeth throughout their lives. Think of like killer whales. Baleen whales, however, only possess teeth in the early fetal stage and lose them before birth. So baleen whales would be something like a humpback whale, where they have those long strands of baleen that they filter food in and out of. And so we can see when baleen whales are in fetal stages, they have teeth, but then those teeth go away and baleen replaces them. But they do still have teeth as a fetus, and so we can see that common ancestry in the same way, where we have a characteristic showing at one point in development, even if it's not seen throughout the individual's entire life. We can also see similarities among living organisms through anatomical structures. So in the context of biology, a homology or a homologous structure is the existence of shared ancestry between a pair of structures or genes in different taxa. So it's something that looks similar, has a similar form and function based on common ancestry and common genes. So a common example of a homologous structure in evolutionary biology are the wings of bats and the arms of primates. So evolutionary theory explains the existence of homologous structures adapted to different purposes as a result of descent with modification from a common ancestor. So all of these tetrapods had a common ancestor that had the same bone structure with an ulna, humerus, a radius, and phalanges, but we have Hox genes which turn on development and change the length of those different structures. So if they're turned on for a longer amount of time, that structure is going to get longer. If they're turned on for a shorter amount of time, the structure is going to be smaller. And so depending, if we look at a human, a dog, a bird, and a whale, depending on the environment that that individual lives, how they need to move, and how they need to interact with their environment, evolution and natural selection have selected for example, in humans, we have 5 digits and we're able to grasp things, whereas in whales, the phalanges, which are the finger bones, are even more extended and they fill out the flippers that help whales steer in the water. But all tetrapod organisms have the same bones in their forelimbs. They're just different proportions depending on the needs of that particular species. So in addition to natural selection, we have what's called artificial selection. And this is where it's not something in an environment that is selecting for different traits. It's actually humans, and in this case, it would be farmers that are selecting for different traits. So different traits in wild mustard have been selected for, resulting in a wide variety of different vegetables. So this wild mustard plant is the ancestor for everything from cauliflower to cabbage to kohlrabi and kale and broccoli and all of that. All of those are the same species and can breed with one another. So you can actually cross kale with broccoli or cauliflower and cabbage because they are the same species. They've just been selected for different things. So in kale, it's been selected for that particular group to be selected for larger leaves. Broccoli has a suppression of flower development. Cabbage is the suppression of internode length. But all of those different vegetables that you see in your grocery store all come from wild mustard, and they have just been artificially selected for different traits. Another example of artificial selection is seen in corn. So the ancestor and close relative of modern corn is called teosinte in North America. And it was over time we selected for more and larger kernels with more and more different types of starches and things. And so we have modern corn, which is the same species as teosinte, and they can still breed together and create a hybrid that looks like a middle ground between teosinte and modern corn. Another one, try to guess what this is. This is a banana. Naturally growing bananas actually look quite a bit different from the bananas that we are used to eating. Naturally growing bananas have large seeds, but through artificial selection, we have selected for bananas to be larger, longer, fuller, and more fleshy with fewer seeds and sweeter taste. And so we have changed the way that bananas looked from the way that they looked in their natural environment to the bananas that humans prefer to eat. The same thing goes for peaches. So we have naturally selected peaches from a very small fruit into these large, fleshy, sugary fruits that have a less waxy peel on them that humans like to consume. So next time we're going to talk more about the mechanisms of selection, and that is the end of our lecture on microevolution.